So hello and welcome to this video on important genetics concepts. This video is a part of our series on genetics made easy. So now let us get into it. So first of all, I want you to know that DNA is made up of, we know this, DNA has chromosomes. These chromosomes are then made up of specific genes. These are at specific points which are known as locus or loci for plural. And then these genes have different versions, right? Some are dominant, some are recessive, some have different traits. So these genes have then something called alleles. These alleles are the versions. So DNA is then divided into chromosomes. Those chromosomes have specific genes. In each gene, you have different versions of that gene called allele. An example of that would be like the color gene has white, red, and blue alleles. So different versions of the same thing is called an allele. So let's say, for the sake of example, white and red are dominant and blue is recessive. Then we talk about the concept of codominance. We know that when one thing is dominant and the other thing is recessive, so what happens is that the dominant thing will take over. For example, if white is dominant and blue is recessive, then the flower definitely will be white, right? And the blue thing will not express itself because that's recessive. What if we talk about a concept called codominance, in which both the white and red express themselves, and then you get a flower that looks something like this, Another example of that is an AB blood group, meaning your, let's say you got the A blood group from your mother, you got the B blood group from your father, and you had codominance. Both of these alleles completely represented themselves, and then you have AB blood group. This is the simplest and easiest example of understanding codominance. Then let's talk about the concept of variable expressivity. Let's say same disease, you have the same disease, but it has separate manifestations, meaning different people have different versions of the same disease. This concept is called variable expressivity. Now, one thing I want you to understand for the concepts that we're going to keep talking about them is that multiple concepts may apply to one single disease. Multiple concepts. It's not just that one disease has codominance and the other has variable expressivity. The same disease can have variable expressivity and also codominance. So the concept of variable expressivity just means that one genetic problem can identify themselves in multiple ways. So let's say this is a particular problem, and then one person it produces A plus B, and in the other person this produces the symptoms of B plus C. So different manifestations in different people. An example of that is neurofibromatosis. In some people you get that combination of symptoms like that cafe au lait spots, neurofibromas. Another person maybe you just get the eye manifestation, those Lish nodules and those spots on the iris, stuff like that. So different people can have different manifestations of the same disease, and we see this pretty commonly in medicine, right? Diseases have variable expressivity. In different people they cause different symptoms. This is the concept of variable expressivity. Then let's say this was the example I talked about earlier. This is someone with NF1. And let's say one person has Lish nodules and neurofibromas and they don't have these symptoms, and the other person, let's say, has skin fold freckling and cafe au lait spots and they don't have these symptoms. So one person gets this and the other person gets this. This is the concept of variable expressivity. Then incomplete penetrance. Incomplete penetrance is very important. What does this mean? That means that even if you do possess, let's say this is the mother, this is the father, and they pass this gene on to, let's say, both their kids, and both of them have this gene, right? So let's say the father is diseased and the mother is diseased, right? And then none of their kids are diseased. Even though this is a genetic hereditary disease, none of their kids have the disease. This is the concept of incomplete penetrance. What does penetrance mean? The word penetrance means whether something passed through, whether something went through. And that just means even though it went through, the disease itself did not pass through. These people, their kids don't have the disease. This is the concept of incomplete or complete. Complete penetrance would be that these people have the disease and then all of their kids have the disease because they have the genetic problem. Therefore, people, even though they possess the genetic problems, the hereditary problems, they don't develop the disease. This is the concept of incomplete penetrance. So the word penetrance means something passing through. And incomplete penetrance, the word means that even though the genetic workup, genetic makeup has passed through, the disease is not there. This is the concept of incomplete penetrance. What's an example for it? The BRCA1 and 2 carriers. Sometimes they develop breast cancer, but in other cases they don't necessarily develop breast cancer. This is a phenomenon that we see for BRCA1 and BRCA2 carriers. So this is the concept of incomplete penetrance. So these people have BRCA1 and BRCA2 genes, but they don't develop breast cancer. Then the concept of pleiotropy. This is a very interesting concept and a highly tested concept that comes up in people's exams. So a single gene has the ability to show multiple manifestations. Now again, I want you to understand that a same disease might be able to show multiple concepts. So a same disease might have, like NF1, if NF1 is occurring due to a problem with a single gene, it can have variable expressivity and also pleiotropy. What is pleiotropy? Pleiotropy just means that, let's say this is a gene, there is a problem with this gene, just this one place, and then what it does is that it causes a plethora of symptoms. It causes multiple symptoms. It causes symptoms in your eyes, it causes trouble on your skin, it causes symptoms of, let's say, tumors in your bones, stuff like that. So a problem with a single gene results in multiple manifestations in multiple body or organ systems. An example of that would be cystic fibrosis, Marfan syndrome. So all of these are problems. Let's say this is a problem in the CFTR gene, and then you know that cystic fibrosis has the ability to affect multiple systems at once. This is the concept of pleiotropy, that you see multiple phenotypes. What is phenotypes? Phenotype is the physical representation of a trait or a gene. That is the physical manifestation, right? So cystic fibrosis is a problem with a single gene, but you see this disease causing problems throughout the body. So a person may have pneumonia, infertility, and stuff like that. And then same goes for Marfan syndromes. Problem with a single gene, multiple manifestation. Another example that we talked about earlier would be NF1. Problem with a single gene may be responsible for causing those Lish nodules, the cafe au lait spots, and everything we talked about. So that's pleiotropy. So this is an example of cystic fibrosis. So cystic fibrosis occurred due to the problem in CFTR gene, a problem with a single gene. And what that would result in is that it causes all of these symptoms: the salty sweat, chronic sinusitis, lung disease, infertility, and particularly in men, and then all of those bones problems. So these are a problem with a single gene is causing all of these multiple manifestations, which is called pleiotropy. How I remember is that the P helps me remember the physical symptoms, the multiple physical symptoms in the concept of pleiotropy. Then let us talk about the concept of anticipation. What is anticipation? Anticipation is a concept particularly seen in trinucleotide repeat diseases. What happens is that there is a combination of nucleotides that are error and faulty, like this, let's say CGG. And what happens is that these three nucleotides start repeating again and again in the DNA, right? They start repeating again and again. And the longer this repetition is, let's say this goes on 25 times in one generation. In the next generation, this thing repeats even more. So it goes on for 35 plus times. And in the next generation of kids, this goes on for 45 plus times. So what happens is the more repetitions you have of this particular trinucleotide repeat, the earlier the disease is going to manifest. So what does that mean? The disease happens earlier and worse with every generation. Every generation. Let's say you started with mother and father started having symptoms of this disease at 55 years. And then you have this son, and this son started having symptoms at 45 years. Then this son has another daughter, and this daughter had symptoms at 35 plus years. This is the concept of anticipation, that diseases are happening earlier and worse with each generation, And why? What is the most common example of this? The most common example of this is trinucleotide repeat diseases. Why? Because the burden of trinucleotide repeats gets increased with every generation. So let's take a look at it. So let's say this is the pennant, and we see this pattern being repeated twice, right? And in their child, we see this pattern being repeated 1, 2, 3, and 4 times. So it's almost double. So let's say this parent had the manifestation of the disease at 55 plus years. Now this kid will have the manifestation just because she possesses a lot of aberrant DNA. She will have at 45 plus. This DNA is not okay. This DNA is wrong. This DNA is bad. We don't want DNA to repeat again and again like this. And then their grandchild possesses the same disease at, let's say, 30 years old. This is the concept of anticipation. What are examples of this? All usually the trinucleotide diseases, particularly like this example is particular for Huntington's disease, in which we have CAG repeat in Huntington's disease. Then how do you remember it? Well, the better physician, in every generation, for example, there are physicians who have multiple generation of physicians, and they keep getting better with each generation. So as a physician, you anticipate better with each generation. As a physician, you anticipate better with each generation. You understand what might be happening because your father was also a doctor and your grandfather was also a doctor, so you were brought and raised in a family of doctors. So as a physician, you anticipate better with each generation what might be happening. Then let us talk about the concept of epistasis. What do you mean by epistasis? So this is pretty cool, right? Like we talked about the white and the red and the blue gene, right? We talked about the white, red and the blue gene. So what happens in epistasis is that there is some other gene right here, which is, let's call it the color gene. And earlier we talked about how the white and red were deciding the color. Now this gene actually, like whatever happens, let's say white and red are inherited from the father. So you get white and you get red. But then this color gene just turns the whole thing black. It turns the whole thing black just because you got a specific allele. So that means there is a third, that there is another gene which is hiding in the background, which is dictating the other gene, which is dictating the behavior of the other gene. Let's take an example for this. What's an example of this? Let's say gene A decides eye color, right? So gene A decides eye color, and then gene B is hiding in the background and it decides melanin production. And if you know about melanin, melanin is the product that causes your eyes to become darker, right? It was actually gene A which was deciding, let's say, light, and you got this combination of light and dark. You got the dark gene from your father, you got the light gene from your mother. So essentially your eyes should be dark because let's say this is dominant, let's say the dark gene is dominant, so your eyes should be dark. But the third gene which is hiding in the background, which decides melanin production, let's say this gene is faulty, or this gene is like no melanin production. So what you will end up having is that you will end up having light eyes, because there is a third gene which is like a switch in the background which is dictating the behavior of the eye color gene. So this is the concept of epistasis, meaning let's say gene A decides the speed of your car, right? Gene A decides the speed of your car. And then there's another gene hiding in the background like gene B, which decides the quality of your engine, which decides the quality. So if the quality of your engine is, let's say, bad, the gene A will be pretty much useless because it is actually B which is deciding whether your car will go or not. So this is the concept of epistasis, that there is another gene hiding in the background which decides whether this gene will express itself or not. The gene B or the bossy gene will dictate the behavior of the other gene. That is the concept of epistasis. Then let us talk about a very interesting concept called linkage disequilibrium. This just means, just remember the word linkage. This means that if you look at this chromosomes and you have these two genes, let's call them A and B. Let's call them A and B. Just because they're together on the same chromosome, these genes have a much higher chance of going together into the next generation, right? That makes sense. They're closer together. They're closer together. Being in a closer together location helps them, even though they're completely unrelated to each other, they're inherited together. And this is the concept of linkage disequilibrium. And why is it important? Well, it's important for us in population genetics. Then we have particular clinical application of this. Let's take an example. For example, socks and shoes drawers are usually together. So wherever you place your shoes, you usually place the socks somewhere close to it, just because you want to understand like where the socks and shoes are. An example of that is that we have seen this association that people with diabetes type 1, we see these two particular genes close together, right? The HLA-DQ2 and HLA-DR3. These things are inherited together. We have seen this linkage that you are more likely to have these two genes together in diabetes type 1. Why? Because we have seen that these two genes are located close to each other in diabetes type 1. You just see them more often being inherited in people with diabetes type 1. So it helps us in clinical understanding of who are the people that might be predisposed to having diabetes type 1, even though we don't understand what the relationship might be, but we just know that gene A and B are located together and they are usually inherited in people with diabetes type 1. That's the concept of linkage disequilibrium. So a very interesting application of linkage disequilibrium is actually in abacavir hypersensitivity. Abacavir is one of the drugs which is used in HIV. It just so happens that some people are hypersensitive to these drugs and we can't use this very good drug in these people. These are the people that usually have this gene, HLA-B5701. We have seen this association. People with this gene are somehow hypersensitive to this drug, which is abacavir. But we usually can't test everyone for HLA-B5701. So how do we do it? We have found a very interesting way of doing it. We found out that on the gene that contains HLA-B5701, there is a very close gene that exists very close to it, right? And they usually exist together. They have no relation to each other. They're just very close together. So they have linkage disequilibrium, right? So they exist very close to each other. Therefore, what we can do is that we can just test for this clinical marker, and most of the time what will happen is that all of these people will have this HLA-B5701 abacavir hypersensitivity gene right here. So we just test for this clinical marker, and more likely or not they will have that particular gene, so we don't give this patient HLA-B57. So another genetic marker which exists very close to HLA-B5701, we just test for that. It's a very interesting concept and interesting use of linkage, of the concept of linkage disequilibrium. So let's talk about a very important concept of mosaicism. Before we go into mosaicism, I want you to understand what is a mosaic. A mosaic is an art piece which contains a random combination of painted or colored objects which is combined into a painting like this picture right here. Some of these are light colored, some of them are dark colored, and you have these distinct colors within the same painting, and these colors are joined together to make this artwork. So this is a mosaic. So what is the concept of mosaicism? It actually is very similar. You have distinct cell lines within the same person. So let's say this was the cell. During division, it is supposed to divide into 2 cells, right? It's supposed to divide into 2 cells. But somewhere along the way, this cell got mutated. And now everything you get from this cell will be mutated, will be blue colored, right? And everything you get from the other one will still be red, I got another color. But everything you got from this, let's say, cell will still be the same color, So this is a very distinct cell line that developed due to a mutation. This is the concept of mosaicism, that after the cell gets fertilized, it generates a new error, right? So what's an example of this? A common example of this is McCune-Albright syndrome, in which you get unilateral spots. Why? Because some cells are being affected during the cell line division and some cells are completely normal. So this is the cell line that is diseased and it passes on to the disease, and this is the cell line which is completely normal. So you get unilateral spots and bone dysplasia that affects specific bones, an endocrine problem that affects specific endocrine glands. Now there is a very important concept in the concept of mosaicism, that there is 2 distinct types. One is somatic mosaicism and one is germline mosaicism. And what's the main difference between the two is that one happens after fertilization, somatic mosaicism happens after fertilization, and germline mosaicism happens before fertilization. How does that happen? So while fertilization, you have this sperm, you have this egg. They join together and they make the zygote, Now after the zygote has formed, you have division into multiple cell lines, and let's say if this one got diseased, and this one is normal, this is the example of somatic mosaicism, that after fertilization you have multiple distinct cell lines in which some of them are diseased. That's the concept of somatic mosaicism. Germline mosaicism is that before fertilization, in the parent's testes or in the parent's ovaries, you develop some sort of mutation, and that mutation is then transferred to the kid. So the parent themselves is unaffected in germline mosaicism. Let's repeat this. So after fertilization in somatic mosaicism, what happens is that in normal body cells and before fertilization in the gonadal cells, so in the testes and the ovaries is where germline mosaicism occurs. So what would be the difference between the two? That somatic mosaicism, although the zygote then develops this diseased cell line and this normal cell line, something like McCune-Albright syndrome, but person with McCune-Albright syndrome will not transfer the disease to their kids. They will not transfer the disease to their kids. Germline mosaicism will develop in the parents or the mothers, basically in their testes and their ovaries, right? And this disease is then transferred to their kids. This disease is then, meaning the person themselves, the parent, will have no symptoms because it particularly developed in their testes or their ovaries, and it just goes on towards their next generation or their kids. And if you talked about their diseased kids, these are the problems which these are the people which will have the problem, not the parents. So in this case, patient or parent has no symptom. Patient or parent has no symptom. In this case, because it happened after fertilization, this person will develop the disease. This person will develop the disease. So this is the example of germline mosaicism because it happens specifically in the gonadal cells of the parent and then it goes on to the kid. Somatic mosaicism, the mutation occurs directly in the patient himself after fertilization. So that's the difference between the two. So example that I talked about earlier is McCune-Albright syndrome. It is an example of somatic mosaicism in which you have unilateral spots, bone dysplasia, and then endocrine issues. Then let us talk about the concept of heteroplasmy. This is a very simple and a very interesting topic, and the best example of this is that the mom carries the mitochondrial DNA. Therefore, mitochondrial diseases are primarily carried from the mom to their kids. So mom is the one that carries the mitochondrial DNA, and this is transferred to her. Let's say if you take an example, this is a mom with a disease, this is a father without the disease, and then all of her kids, no matter how many kids she has, will have the disease just because the mom has the disease. But let's say her diseased son marries a normal mom, so his kids will be normal because only the mom can transfer the disease to their offspring. This is the concept of mitochondrial DNA, which is passed on to her kids. Now, an important concept is the amount of faulty mitochondrial DNA that someone gets, that one kid gets, actually determines the disease severity. An example of this is basically Melas syndrome, in which you have mitochondrial encephalopathy and lactic acidosis, and then Leber's hereditary optic neuropathy, which is problem in the person's eye, right? So these are example of mitochondrial diseases. That means, let's say your mom has these mitochondria, right? Your mom has 4 mitochondria, 3 of them are diseased, right? 3 of them are diseased. And you just happen to be the kid who got the normal mitochondria, so you might be completely normal. This is an example of heteroplasmy, in which the amount of diseased DNA you get from your mom actually determines the disease severity. Let's take an example like this. Let's say this is the disease. Let's say this is the disease containing mitochondrial DNA that you got from this is the mom, right? This mom passes on some mitochondrial DNA to her kids, which is completely normal, right? So this actually develops a kid that is completely normal, and then some mitochondrial DNA which is faulty like this, which then replicates like this, and then some kids might end up completely having this DNA. Some kids might end up completely having DNA, and the amount of this faulty DNA is what is going to determine whether this disease is going to be severe or not. For example, if we talk about this person, then this person might have some disease severity, not a lot, but some disease severity. This person will not have the disease at all, no disease at all. This person will completely normal. This person will be very, very diseased. This person will be very, very diseased. So this is the concept of heteroplasmy. Let's take another example like this. So this is a bottle. It contains some faulty mitochondria. If you look here, some faulty mitochondrial DNA. So some people will end up getting a plate like this, which has no faulty mitochondria. It's like mom is getting you food from the main container. Some of the food is poisoned, a lot of it is not poisoned. The person who will eat the most poisoned food will actually get the worst disease. The person who will eat the least poisoned food will get the least disease, right? So you get this person who's completely normal because they were just lucky to not have any of the disease genes. They were very unlucky to have a lot of the disease genes. So if you look at this person, right, if you look at this person, they actually end up having a lot of the disease. This person is completely normal. This person is completely diseased. This person is somewhere in the middle. This person is somewhere in the middle. So amount of diseased DNA that you get from your mother's pot is going to decide what is the severity of the disease that you're going to get. This is the concept of heteroplasmy. Then let us talk about the concept of locus heterogeneity, which is mutation in different locations, different genes. Mutations in different locations and different genes end up resulting in the same disease, What's an example for this? Example for this is osteogenesis imperfecta. We know that there are multiple genes associated with it, like COL1A, COL2A if I'm not wrong, and all of these are associated with osteogenesis imperfecta. So there are mutations in different locations, in different gene. Locus, from locus you can remember the fact that this is talking about location. They're talking about the location. So mutations in different locations can still cause the same disease. An example of that is osteogenesis imperfecta, retinitis pigmentosa. Mutations in different locations causing the same disease. Then we have the concept of allelic heterogeneity. And allelic heterogeneity and how it differs from locus heterogeneity is that it's mutation in the same gene, in the same location. Mutation in the same gene or same location results in the same disease. What it happens is, let's say you had this gene which decided eye color, right? And now you're getting strange mutations in it which is causing your eye color to be gray, or whatever kind of deletion is that is causing your eye color to be black. Now this is an example of allelic heterogeneity in which you have different kinds of mutation happening in the same location resulting in the same disease. So let's put them side by side so we maybe understand them better. So locus heterogeneity and then allelic heterogeneity. So locus heterogeneity is different locations, different genes can result in the same disease. An example I talked about was osteogenesis imperfecta. And allelic heterogeneity is that problems in the same gene in the same location can result in the same, so multiple problems, whatever the problem might be in the same gene, same location results in the same disease. That an example of that is beta thalassemia. We know that there are multiple different types of beta thalassemia mutations which occurred in that particular gene, and they all result in different variants of the disease or the same disease, which is beta thalassemia. So that is an example of allelic heterogeneity. The different alleles of the same gene, different type of allele have the potential to cause the same disease. That is the concept of allelic heterogeneity. Taking an example for it, so the difference between locus heterogeneity and allelic heterogeneity can be understood with the example, so that you have problems anywhere in the engine, so that on different locations in the engine, but all of them actually result in the car not starting, And allelic heterogeneity, what happens is that you have very specific spark plug problems, very specific different kinds of problem with the spark plug itself, and what it causes is that car still doesn't start. So you have different problems in the same location, same gene, same different kind of allelic mutations, deletion, whatever that mutation might be, and the problem in the end is the same, which is like beta thalassemia or the car not starting. Then locus heterogeneity, problems at different locations in the engine, but the disease itself is still that the car doesn't start. So that's the difference between the two. So what's the main takeaways from these lectures? DNA is made up of chromosomes which has genes. Those genes have different versions called alleles. Dominant genes mask the recessive genes. Codominant genes both express themselves, like you get the A blood group from your mom, B blood group from your dad, and then you have AB blood group. Variable expressivity, what does variable expressivity means? You have same disease, separate manifestations, and we talked about the example of NF or neurofibromatosis, in which different people can have different manifestations even though they have the same genetic makeup, right? Incomplete penetrance, penetrance means passing through something even though you have the genetic makeup for having the disease, but you don't express the disease. That is the example of incomplete penetrance, which is that some people don't develop the disease at all. Then pleiotropy. Pleiotropy is that one gene, it's just like we're fascinated that one gene can be responsible for multiple phenotypic presentations, that multiple physical manifestations of a single disease, like the cystic fibrosis we talked about. It's a problem in the symbol CFTR gene, and what it results is that you cause a plethora of problems all throughout the body. Talking about the concept of anticipation, with each generation you anticipate the disease earlier. That is, the disease happens earlier with each generation and worse. Why? Because the trinucleotide repeat, we talked about trinucleotide repeat disorders, is getting higher and higher. The ratio of those repeats is getting higher and higher. So an example of that is that Huntington's disease. It happens earlier with each generation. With the father, if it was 55, then in the kid it's 45, and then it might happen at 35 in the next generation, and even worse. Then the concept of epistasis, that there's another bossy gene that is hanging in the background, and that is actually deciding whether this gene expresses themselves or not. So there's another gene which dictates the activity of one gene, let's say. So, and then we talked about the concept of linkage disequilibrium. So just for an example of epistasis is the eye color and melanin production gene that I talked about. For linkage disequilibrium, just because two genes might be located together, they might be neighbors of each other, that just means that there is a higher probability of them existing together, and then we use that to our advantage. Genes closer together are inherited together, or maybe linkage disequilibrium can maybe work the other way as well. The genes far from each other are actually inherited worse, like they're not inherited usually together, so it can work in both ways. Then mosaicism, which is distinct cell lines within the same person. If it happens after fertilization and the person becomes diseased, that's somatic mosaicism. If it happens before fertilization in the moms and pops, testes and ovaries, and therefore that's germline or gonadal mosaicism. Then heteroplasmy, in which the amount of faulty DNA that you get from your mother actually determines whether you develop the disease or not. An example of that is definitely mitochondrial diseases like MELAS, and the amount of faulty DNA you get from the mother's pot will actually determine whether you have the disease or not. That is the concept of heteroplasmy. So putting locus heterogeneity and allelic heterogeneity side by side, so different genes, different location, problem anywhere in the engine causing your car to not start, so different genes and different location causing the same disease, an example of locus heterogeneity. And same gene, same location, problem with the spark plug, whatever the problem might be, you can have all different sorts of mutations, deletions, whatever is happening with the spark plug in the same location. What it happens is that you develop the same disease, that the car doesn't start. That is the difference between locus heterogeneity and allelic heterogeneity. That was pretty much all I had to talk about for concepts in genetics. Thank you so much for watching this video. This video is a part of our series on genetics. And if you haven't watched my series on biostats and epidemiology, be sure to watch that. Thank you.